Welcome to the biology class. In this video, we are going to start a new lesson. Unit 16, Plant and Animal Hormones. We are going to see introduction and the first plant hormones auxins in this video. Hormones are chemicals. These chemicals carry information or messages. These hormones are signaling molecules. In the last lesson, we learned that the nerves carry the signals. Hormones also carry signals. They carry information. So they are called as chemical messengers. These chemical messengers regulate physiological activities. They control metabolic activities. They control biochemical activities. They maintain coordination among various functioning of the organisms. Plant hormones are called as phytohormones. Plant hormones are chemicals produced by a particular part of a plant. They diffuse into other parts in very low concentration. These hormones promote physiological activities. In plants, several cells are capable of producing hormones. Plants do not have separate organs for hormone production. Several cells have the capacity to produce hormones. In animals, the hormones are produced by glands called as endocrine glands. Our body has various endocrine glands. All these glands form endocrine system. We have adrenal gland, pituitary gland, pineal gland, various glands we have. These glands form a system, endocrine system. These glands produce hormones. The hormones are transported by the blood. Endocrine glands are ductless glands. They do not have a tube to carry their secretions. Their secretions are transported by the blood. In plants, the cells are capable of producing hormones. In animals, there is separate system with endocrine glands to produce hormones. In this video, we start the phytohormones. Auxins, cytokinins, gibberellins, abscisic acid, ethylene are various phytohormones. The first three hormones promote plant growth. Auxins, cytokinins, gibberellins promote plant growth. Abscisic acid and ethylene inhibit plant growth. Are the first plant hormones to be discovered. Auxins are produced at the tip of stems and roots. They are produced at the tip of stem and roots and they diffuse into other areas where they do the physiological functioning. Charles Darwin in the year 1880 observed a kind of growth in canary grass. The plants show bending movement. They bent on one side. He observed unilateral growth and curvature of canary grass. Unilateral growth on one side. It is a curvature growth, that is bending growth. In a plant, The tip is intact. The tip is not damaged. In monocot plants, the young plant tip is covered by coleoptile. Coleoptile is a covering. It covers tip of young plants. Here the coleoptile is undamaged. So it shows bending movement when it is exposed to the light. It bends towards the direction of light. In the second case, the tip is damaged, the tip is removed, it does not show bending movement. On seeing this observation, he came to the conclusion. That some influence was transmitted from the tip of the coleoptile to the basal region. From the tip to basal region, something is transmitted that caused the bending. That was his conclusion. The influence was later identified as Auxins by Wendt. Auxins were first identified by Wendt. Conducted experiment on Avena coleoptile. He conducted coleoptile experiment on Avena plant. Avena plant is oats plant. Here the picture shows the presence of coleoptile. Inside the coleoptile there is a young leaf. Coleoptile covers the growing tip of monocot plants. He removed the tip of coleoptile. He removed the tip of Avena coleoptile. In another experiment, he placed the agar block on the decapitated coleoptile tips. Agar block is a small piece of agar that was placed on the cut end of coleoptile. The agar block was kept on decapitated coleoptile. He didn't find any change. The coleoptile did not show any response. The coleoptile did not grow. In his next experiment, he placed the detached coleoptile tips on agar blocks. Look at this picture. Here there is a detached coleoptile tip, a cut tip. This tip was kept on agar block, a small piece of agar on which the detached coleoptile tip was kept. Now what is possible? The chemicals inside the tip can spread into the agar block. After an hour, he discarded the tips and placed this agar block on the decapitated coleoptile. He didn't use the tip for further experiment. He continued the experiment with agar block. He kept the agar block on the decapitated coleoptile. The agar block was kept on the decapitated coleoptile. He observed growth. The coleoptile elongated. What may be the reason? The chemical inside the tip of the coleoptile diffused into the agar block. While placing the agar block on the cut end, these chemicals entered into the plant and promoted the growth. He named this chemical as Auxin. The meaning of the Auxin is to grow. This is the discovery of Auxin. There are natural Auxins and synthetic Auxins. Natural Auxins are produced by the plants. Example IAA, indole 3 acetic acid. Synthetic Auxins are 2,4-dichlorophenoxyacetic acid. PAA, IAN are other examples of natural Auxins. Phenyl acetic acid and indole 3 acetonitrile. Indole 3 butyric acid, indole 3 propionic acid, alpha naphthalene acetic acid, 2,4,5 trichlorophenoxyacetic acid are synthetic auxins. Auxins promote growth. Auxins are produced at the tip of the plant. From the tips, the auxins spread downwards and promote growth. Auxins promote the elongation of stem and coleoptile. Auxins make coleoptile and stem to grow. Auxins promote root formation in low concentration. Auxins induce root formation at low concentration and inhibit root formation at higher concentration. Low concentration of auxins induce root formation. The auxins produced with the apical buds suppress the growth of lateral buds. In this picture, the apical bud is undamaged. The auxin produced by the apical buds spread downwards and suppress the growth of lateral buds. In this picture, the lateral bud growth is suppressed. Lateral buds do not grow. Look at this picture. Here, the apical bud is damaged. So, auxin cannot be produced. This auxin cannot be produced. As a result of this, the lateral buds growth takes place. In all the areas, the lateral buds grow well. This is called as apical dominance. Auxins produced by the apical buds spread downwards and suppress the growth of lateral buds. This is called as apical dominance. If the apical bud is removed, then lateral buds grow well. Auxins promote seedless fruits. Seedless fruits are produced without fertilization. Seedless fruits without fertilization are induced by the external application of auxins. This is called as parthenocarpy. Seedless fruits are called as parthenocarpic fruits. They are produced by spraying auxins on the flowers. Example: watermelon, grapes and lime. Auxins prevent the formation of abscission layer. Abscission layer is a layer of loosely arranged cells. Abscission layer leads to abscission. Falling of fruits and flowers is called as abscission. Abscission layer is formed at the base of the leaf. This layer has loosely arranged cells. As a result of this, the leaf falls down. Auxins prevent abscission. When auxins are there, the young leaves do not fall down. Young flowers and young fruits do not fall down. So auxins prevent abscission.